49. Like I said, we got 10 minutes to do this. Olá. Buenos dias. ¿Cómo estás? What is this? Morning. I don't know Spanish for I hope you all right. I'm trying to think how to say tired. There we go. ¿Cómo se dice tired? Cansado?
Cansado.
Cansado. Gracias. Just to answer your question. Muy cansado. All right, it's 8 o'clock. Let me just do attendance. If you have the state gear on, let me know when I call you in. Brumfield, here. Champion is here. WBSU here. Gorby, I think, is online. Maybe Gross is online. Gunther is here. Yeah, WBSU stuff on? Yep. All right. Randall is here. Got your WBSU stuff too? All right, there we go. So both of those are here. Jackson? Jackson is not here. Keys is here, and WBSU here. Nez is here, and WBSU here. Sigmund is here in WBSU gear, right? Yeah. Okay. Slaughter is not here. Smith is here back there, I think, right? Yep. Stover is here. Her jacket's shirt. Tompkins is here. Wait, no, it's not. Tompkins is not here. Zanest is here. Watson here. Watson's not here. Young is here. Zweiback is here. Lawrence is not here. Wilson is not here. And that's it. Did I miss anybody? So obviously, again, attendance is mandatory. It's graded. You guys know what to do if you're in here. If you're watching the video, I guess technically it is a possibility. This is the first video you've watched. So every time I say an attendance word or an attendance phrase, you're going to write it down. You're also going to take a screenshot and send it to me as proof that you watched the video. The last word of attendance or phrase for attendance is going to be you asking me a question about the material presented in this lecture. So any questions about attendance? All right. The first word for attendance is breast. Because today is National Dense Breast Awareness Day, so it's a good reminder. Get your mammograms, get your self-testing. Any questions about that? All right. So some reminders again. Make sure you're paying attention because I've been saying this for a while, and some people still didn't even understand it until recently. You can still retake the first exam, even if you never took it in the first place. Keep retaking it as many times as you want. It's open book. It's not timed. The only catch is once exam 2 is done, then you can't retake exam 1 anymore. And it's important because people forget this. You have to let me know when you're done taking it. Otherwise, I will not know to change your grade. Let me know once you're done taking the exam. You have nothing to lose. If somehow you keep getting a lower grade, which I don't see how because it's not timed and it's open book. But if you somehow keep getting a lower grade, you just won't change your grade, but keep your higher grade. So I recommend doing that, especially if you're the kind of student who you know and you know yourself. It's going to be the end of the semester and you say, hey, is there anything I can do about my grade? I'll say, these are the kind of things I'm going to be looking at. Like, well, did you retake the exams? Did you do any extra credit? Things like that. So go ahead and get on it. Let me know if you need help. Any questions about the exam? All right, lab is today at 9 a.m. as usual. The only difference is it's just a pre-lab lecture, right? So I'm going to be going into full detail about how these experiments are going to work next week because that way we can hit the ground running once we get here next week. And because you have to write a lab report about it, it's even more important that you really, really, really understand the concepts behind these experiments. So also I'll be giving a little bit of a lecture on how to write a lab report. All that's going to happen today. So your grade is based on being here on time. All you have to do is be here on time, and not leave early, and you get your full 20 points. Come in late, you're going to get partial credit. If you leave early, you get partial credit. Obviously, if you're not here at all, you get 0 credit. So, any questions about lab today? All right, again, pre-labs due this Friday as usual. So just as a heads up, it's a 3-section, 3-part pre-lab. So there's 2 videos with a set of questions for those. Then you have the introduction, and then the pre-lab questions to prove that you read the introduction, so make sure your answers come from the introduction. And then, of course, you'll have the hypothesis questions. So there'll be 5 sets of hypothesis questions because this lab consists of 5 different experiments. So you'll need to have a hypothesis for each one of those experiments. And again, you don't have to do these alone. These aren't quizzes, these aren't tests. So if you need help doing the pre-lab between now and Friday, let me know. Maybe online, I'd be happy to help. So any questions about announcements and reminders? Do any of you guys have any announcements or reminders? All right, what time's I don't go to the game to 1:30. 1:30. 1 o'clock? Okay. Anyway, as usual, if you go to that or any other official WBSU event and you have proof it with pictures there, where I can tell it's you, your face and everything, you get extra credit. Hopefully I'll see you there. Anyway, let's move forward. So to remind you, we were talking about respiration, right? Actually, let me back up even more. Obviously, what we're doing is covering material that's going to be on exam 2. almost the entirety of exam 2 was all about the property of life of energy, right? How we have to change energy from one form to another. That's the property of life. That's almost what this entire exam is about. I was telling you before we started, during this chapter, I said one of the most important things you need to know is this will be a test question, and everything else is just small details. If you don't remember this one important detail, your cells need ATP for energy. Right? That's where they get their energy. That is their direct energy currency. So to get ATP, we break down break down glucose in the presence of oxygen, right? That is what cellular respiration is, and that's what we're talking about. Cellular respiration is broken down into 3 stages. There's glycolysis, citric acid cycle, and the electron transport chain / oxidative phosphorylation. Same thing, different names. We've already talked about the first 2, and we've even started talking about electron transport chain. Right? I mentioned one of the important things you need to know about this, or the whole big picture, is these high energy electrons are embedded in that glucose. So as we break down that glucose in the first 2 stages, we're stripping away these high energy electrons. And then those high energy electrons are transported to the mitochondria, where finally they are put to use. And they go into the mitochondria, there's these proteins embedded in the mitochondria, and as they go from one protein to the next, they're releasing their energy. That energy is used to pump hydrogen ions against the concentration gradient. Because, when you go against the concentration gradient, that is active transport, right? So you need energy to go against the concentration gradient. So that's what's happening. That release of energy from the protein the electrons going from protein to protein is used to pump the hydrogen ions against the concentration gradient. There they just do what they naturally would do. Nature takes its place, and then the hydrogens diffuse back down the concentration gradient. And the only way they can make it through the membrane is through this enzyme called ATP synthase. Right? And ATP synthase, as they're going through, it literally turns, and that's where we make the ATP. So again, there's a lot of test questions on this one slide. So which of the 3 stages, pumps hydrogen ions against the concentration gradient? Electron transport chain. Which of the 3 stages produces ATP by letting hydrogen ions flow back down the concentration gradient? Electron transport chain. Here's another test question. I don't know if I've mentioned this as a test question. What is the enzyme responsible for making ATP in the electron transport chain? ATP synthase. Yeah, that's supposed to be it. But yeah, which of the 3 stages uses oxygen? Electron transport chain. Which of the 3 stages produces water? Electron transport chain. So yeah, that's pretty much it. Now there's a lot of other details on here that we're not necessarily going to get into. For example, you can see here, we've talked about NADH, we've talked about FADH2, and I told you as far as you're concerned, that's the same molecule, right? They're just electron carriers. But this picture from the old textbook, they get even more detail. Technically, yes, there's 3 proteins involved in this process. And technically, the NADH takes the proteins to the electrons to the first protein. And technically, the FADH2 was the one that brings it to the second protein, but you don't need to know all that. I'm not going to get into that much detail. So yeah. But yeah, you see this little skull and crossbones. That's kind of like a little indicator of this is how not breathing kills you, right? This is how drowning kills you. But you don't get enough oxygen. There's nowhere for these electrons to go. And if there's nowhere for the electrons to go, just like in an electrical circuit, you have plugged up both sides. If they can't flow, they don't flow. If that doesn't happen, you don't make ATP. And if you don't make ATP, die. So that's it for the 3 stages of respiration. Any questions about this slide? All right, so some numbers you need to know. And I mentioned it before, so some of this is a review. Cellular respiration generates about 32 ATP per molecule of glucose. You need to know that number. Now, if you look at other sources, sometimes it says 38. Don't worry, I'm not going to be tricky and have both of those options on your multiple choice exam. It's either going to be 32 or 38. So anything in the 30s is the correct answer. Anything not in the 30s is not the correct answer. So even though I'm not going to get that detail in the exam, the next slide is going to help us keep track. We're going to review where all these ATP were generated. And it'll also be another good review for the exam, because I'm going to give you some exam questions again. Any questions about this slide? All right. Here's a picture from the old textbook. There's so many test questions on here. Like, which of the 3 stages of respiration happens outside of the mitochondria? Glycolysis. Which of the 3 stages happens inside of the mitochondria? Citric acid cycle and electron transport chain. Which of the three stages directly uses glucose? Glycolysis. Which one produces pyruvate, aka pyruvic acid? They don't say pyruvate on the exam. Glycolysis. Which of the two stages is associated with acetyl-CoA? Citric acid cycle. Which of the three stages produces carbon dioxide, even though it's not on this slide? Carbon dioxide. Excuse me, citric acid cycle. Which of the three stages produces ATP? All three. Which of the three stages produces the most ATP? Electron transport chain. We're almost there. Which of the three stages strips away high energy electrons? Glycolysis and citric acid cycles. And which one uses high energy electrons? Electron transport chain. That's also a commonly missed question, so make sure you read that question carefully. One question is going to say which of the three stages strips away high energy electrons, right? And that's the first two. And there'll be another one that says which of the one uses high energy electrons. Hopefully that one's easy to remember because it's called the electron transport chain for a reason. Anyway, any questions about that picture? Okay, so like I mentioned, electrons have to come from somewhere, they have to go somewhere. Where do they come from? As far as we're concerned, it's glucose. As far as the exam is concerned, it's definitely glucose. Here in a few minutes, we're going to talk about some exceptions of that. Where do the electrons go? They go to the oxygen. That produces water. That's definitely going to be a test question. Come from glucose. They're accepted by oxygen, which produces water. That will be a test question where you have to fill in the blank. There'll be a whole little sentence or paragraph. That's the order, right? The electrons come from glucose. They're accepted by oxygen. That produces water. Now, like I said in the previous slide, this is what kills you, right? This is how not having oxygen kills you, is because if your electrons don't have somewhere to flow, they don't flow. If they're not flowing, you're not producing ATP. So let's talk about some exceptions to that rule. You guys have probably heard of this term before, but it's fermentation. So let's talk about fermentation. Here's the objectives. By the end of this section, you should be able to discuss the fundamental differences between anaerobic cellular respiration and fermentation. For the exam, there's not going to be much difference between the two, but you'll see there are actually some fundamental differences. And you should be able to describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So let's get started. So cells can work a little bit without oxygen, right? So, if you hold your breath, generally speaking, you don't die, right? If you just hold your breath a little bit and you're in the pool, want to see how far you can swim, you can go a little bit without oxygen. And that's where fermentation comes in. Fermentation is the anaerobic harvest of food energy. And just like cellular respiration, probably not going to ask you the definition of it. I just want you to know how it works. So even though I just gave you the definition of fermentation, the more important thing here is you understand how it works. And that's what I'm about to teach you. And when I said anaerobic harvest of food energy, what I mean is without oxygen. So basically what I'm saying is we're still breaking down food. And again, just to keep things simple for this 100 level course, we're just going to focus on glucose, but there are other things. We are breaking down glucose, but we're doing it without oxygen. And that is fermentation. Any questions so far? Okay. After functioning anaerobically for about 15 seconds, and that's not a number you need to know, just let you know that's the average, muscle cells will start generating ATP by using fermentation. Fermentation relies on glycolysis only. You do need to know that. So, you just learned about all these three stages of cellular respiration. But when it comes to fermentation, it only uses glycolysis. Why is that? Because glycolysis does not directly require oxygen. Right? You know that the only one that directly requires oxygen is electron transport chain. Glycolysis does not directly. Neither, again, come to think of it, neither does citric acid cycle. So at the end of this, I'm going to ask you guys to guess, why is it that it's only glycolysis and not the citric acid cycle? I don't expect you to be able to guess it now, but a little bit later, you should have some more information, and let's see if anybody can figure it out. Anyway, as you know previously, excuse me, glycolysis produces, or nets, 2 ATP per glucose. I say nets, remember, because technically it produces 4 ATP, but you have to use 2 to get the whole thing started, so that means that, profit is 2 ATP. But yeah, so definitely know that fermentation relies on glycolysis, right? Because glycolysis does not require oxygen directly. So any questions so far? All right, as a reminder, NAD+, that's that thing, right? That's that bucket. They usually call it a bucket. That's that bucket that's carrying the electrons from one place to another. When it has a plus, that means it's an empty bucket, right? It's ready to accept those electrons. So you need those buckets, right? You need those empty buckets to take those electrons so we can strip away the electrons. Because remember, glycolysis, for the most part, that's how we were making the ATP, was by stripping away those electrons. And even though we don't need to go into this much detail for the exam, if you remember when we were stripping away those electrons, we were also adding phosphate groups to the pyruvate and then taking those phosphate groups back away. All those details you don't need to know for the exam. But yeah, the point is, the whole process of cellular respiration, which we're done talking about, that requires oxygen. What this requires, instead of oxygen, is NAD+, the empty buckets. Right? And that's not a problem in aerobic conditions, right? If you have oxygen, that's not a problem because those NAD pluses, they take the electrons, they take them to the electron transport chain, they dump out the electrons, so to speak, now they're empty again, they go back to glycolysis and they're ready to do the job. But we don't have oxygen, then the question is, where are we gonna put the electrons, right? Those electrons have to go somewhere. We said that in the last lecture. Electrons have to go somewhere. So where are they going? We'll get to that in a second. The next slide I'm going to show you is kind of a reminder of how things are supposed to work, but how they don't work without oxygen. So basically a visualization of this PowerPoint. But are there any questions about this slide? Are there any questions about this slide? All right, let's move forward. So this is the picture you already saw, right? So this is how it normally works when we have oxygen, when we're doing cellular respiration, right? We strip away these high energy electrons. They're taken to the electron transport chain. They go from protein to protein, releasing their energy. We use hydrogen ions to make some ATP. Oxygen eventually accepts the electrons and becomes water. But here, since we're talking about fermentation, again, we don't have the oxygen. So this can't happen. We've got these NADH, these electrons, and nowhere to put them. So we have to recycle them. Right? Without the electrons, the oxygen to accept the electrons, we have to have somewhere else to put them. So instead, and I should have asked if you guys could have guessed this. Hopefully somebody could have eventually guessed it. The electrons are then added to the pyruvate. The reason I'm saying hopefully somebody could have guessed it is because if you remember about glycolysis, for the most part, the only thing I talked to you is that we started with glucose, and we broke it down into pyruvate. In the process, we stripped away high energy electrons and made some ATP. But since I told you that's the only thing we produce, right, that's the only molecules that we're making in glycolysis, pyruvate. So hopefully you could have guessed, after a long wait maybe, that that's where the electrons have to go. That's the only place, the only thing I've talked to you about. So anyway, yes, we strip away these high energy electrons. Can't bring them to the oxygen because there is no oxygen. So instead, we add them to the pyruvate. Once you add those electrons to the pyruvate, then the NAD plus is ready to go again, right? You have your empty bucket ready to get more electrons. And that is what keeps glycolysis working in this anaerobic situation. Here's some more information. This one's important for the exam. When you add, there we go. It's hard to work with. When you add those electrons to pyruvate, it now changes it, right? Every time you do something to a molecule, change it, right? If you add electrons, take away electrons, it technically has a different name, right? It's no different here. When we add these electrons, we then produce lactic acid, and that will be a test question. What does fermentation produce? One of the answers is lactic acid. I'll tell you about another one here pretty soon. But the test question will get more specific and say something like, in animal cells, what does fermentation produce? Lactic acid. And one way, I'll go back to that on the next slide. Anyway, not that you need to know this for the exam, but that lactic acid is then transported to the liver, where the liver converts it back to pyruvate. Pyruvate goes back to the cells, and the whole process continues. But of course, again, this is fermentation, so for humans, you can't do this long, we'll die without oxygen, but It keeps us alive for a little bit. The next slide I'm going to show you is basically a visualization of what's on this slide. So are there any questions about this slide? All right. So here's what I was about to mention earlier, but I wanted to wait until there's a picture. So if you've ever run really fast or done anything where it's like really, you're breathing heavy and you're out of breath, maybe you feel a burn in your chest, maybe you feel a burn in your legs, wherever the burn may be, that's part of the reason why. Because you've used all the oxygen, right? You're working out so hard that you're using all your oxygen, therefore you start to go into fermentation. When that happens, it produces lactic acid. And acid's got to burn, so that's where you get the burn from. One of the reasons you get the burn. Anyway, that's not important for the test. I just wanted to mention that because hopefully that'll help you remember up for the exam. When I ask what does fermentation produce in animal cells, the answer is lactic acid. With that being said, here's just a visualization of what we said earlier. Again, this is glycolysis. So we start with glucose. We break that down in half so you make 2 pyruvate molecules. Again, I like to say pyruvate instead of pyruvic acid. We talked about that previously. In the process, we strip away these high energy electrons. We've made some ATP. Now these high energy electrons have nowhere to go, right? Because there's no oxygen. So instead, they're added back to the pyruvate, which produces lactic acid. That's how it works. I think this slide's better than the next one I'm going to show you. This is from the old textbook. The one I'm going to show you next is from, oh wait, never mind. It's an animation. Excuse me. But are there any questions about this slide? All right. So if you download the PowerPoint, you can watch this little animation, but it's basically everything we just said. And then some. So some of this stuff I haven't taught you yet. So here's what I did teach you. Break down glucose. We've stripped away high energy electrons. Those things are added back to the pyruvate, which produces lactate, aka lactic acid. What I haven't taught you is this. Yeast can also produce ethanol, which is basically alcohol. Well, it is. It's alcohol. The kind of stuff you get in your beer and your wine and your liquor, that's what yeast produces. I think that's what most people think of when they think of fermentation. But again, I haven't even talked to you that yet. We'll get there in a second. So any questions? All right. So your book, your old textbook points out, right? So we just got finished talking about cellular respiration in animal cells, right? Human cells. And we know, obviously common sense should tell you, we cannot survive without oxygen very long, right? We do fermentation for a little bit, but it's not sustainable. However, if we're talking about things other than animals, like microorganisms, fermentation can do the trick. And we're going to specifically talk about yeast. And I'm going to list a bunch of stuff that's not in your textbook, and I'm not going to test you on it. It's just to hopefully make you familiar. You'll be like, oh yeah, I recognize that stuff. So now you know where it comes from. It comes from the lactic acid fermentation of yeast. Produces cheese, sour cream, yogurt, soy sauce, pickles, cabbage, olives, sauces, meat products, probably other stuff too. Again, you don't need to know any of that. The point is that we can use lactic acid fermentation with yeast, right? We can make yeast produce lactic acid. And in doing so, we can make them produce these things. Now, if the conditions are right, we can also have yeast create other stuff. Actually, before I talk about that, let me mention this. Yeast is a microorganism, right? Yeast is a single-cell organism. But it's a little bit different than a lot of the single-cell organisms you might be familiar with. Like me, when I think of single-cell organisms, I think of bacteria. You remember from previous discussions, bacteria are prokaryotes, right? It's very basic life forms. Yeast are eukaryotes, right? They're a type of fungi. So they're just, I won't say just like us, but we are eukaryotes, right? So plants, animals, fungi, those are eukaryotes. So they, even though they're single cell, they are still a little bit more complicated. And because of that, they can still do regular cellular respiration, right? So if they're in the presence of oxygen, they'll do cellular respiration just like I talked to you before I talked to you about fermentation. But if you get the conditions just right, you deprive it of oxygen, then you can start getting it to do alcoholic fermentation. And obviously, you don't need to know this for the exam, but if you want to look it up for extra credit, how do people do that, right? Like, we use yeast, again, like I mentioned earlier, to do lactic acid fermentation, which can do things like make sausages. But we can also have it do alcoholic fermentation. So you can ask yourself that, or look that up for extra credit. How is it that we do that? What do we do to yeast to make it make alcohol versus lactic acid versus cellular respiration, right? So it has three different options: regular old cellular respiration, fermentation where it produces alcohol, or fermentation where it produces lactic acid. But anyway, yeah, so obviously I'm not going to ask you about beer and wine or liquor, but yeah, the yeast is obviously used to make that kind of stuff with alcohol. Like, for example, if you've ever seen people make bread from scratch, it causes the dough to rise because this whole process also produces carbon dioxide. Anyway, any questions so far? Right. I'm probably not going to ask you any questions about yeast. I'm just giving you a little bit of background. So this should look very familiar. It's different, but familiar. So again, this is still fermentation, but we're not talking about lactic acid fermentation. We're talking about alcohol fermentation. So we're still starting off with glucose. We're still breaking it down in half into pyruvate. We're still stripping away high energy electrons. Those high energy electrons still can't go to oxygen because we don't have any, so they have to be added somewhere else. So in this case, they're added back to the pyruvic acid, aka pyruvate. But in this case, instead of turning into lactic acid, which is a 3-carbon molecule, it turns into ethanol or ethyl alcohol, whatever you want to call it. And in the process, again, since these are 3-carbon molecules, or lactic acid is 3-carbon, right? These are 3-carbon. So obviously, if we're going to go from a 3-carbon molecule to a 2-carbon molecule, we're going to lose a carbon, and that's what the CO2 comes from. Now, I've already mentioned this previously. There's going to be questions on the exam that says, which of the 3 stages produces carbon dioxide? Don't worry, for this multiple choice exam, you're not going to have to remember, okay, there was one type of fermentation that produces alcohol. But then there was also the citric acid cycle that produces carbon dioxide, right? So if you get a question about carbon dioxide, it'll be straightforward. If it's about cellular respiration as opposed to fermentation, then your only options are going to be glycolysis, citric acid cycle, or electron transport. Anyway, yeah, and again, for extra credit, because you don't need to know this for the exam, but what causes this? I mean, if you're looking at this picture and thinking about the other picture I showed you, why? What is it about what is it about this process that makes us lose 2 carbon dioxides? As opposed to keeping them, losing 2 carbons, as opposed to keeping them and producing lactic acid instead of ethanol. So you can look that up for the exam if you want. And as a reminder, like I said a few times in the previous discussion, we really oversimplified this. And you learned about enzymes, and I told you there's a specific enzyme for every chemical reaction that happens in your body. So what are the enzymes responsible for all these different chemical reactions? And also keep in mind this is simplified, so there are other chemical reactions between here and here, between here and here. They're just not being shown in this simplified picture. Any questions about that? All right. So some prokaryotes, which I just mentioned previously, right? So we're talking about bacteria and some archaea. They can also use anaerobic respiration. As a matter of fact, some of them, they only live in anaerobic conditions. Some of them. Other ones require oxygen, some don't. I'm not going to test you on this. This is just for your own information. So some examples of some methanogens. And they break carbon dioxide down to make methane. So there's terms I didn't teach you yet because I'm not going to use them on the exam. Oxidizing is when you take away electrons from something. This was always confusing to me when I was like a freshman. Because that part makes sense. Oxidize, take away electrons. Sure, why not? That's not complicated. What used to throw me a little bit was when you reduce something, you're adding electrons to it, which I always hate because if you reduce, it sounds like you're taking away something, but in this case, you're not. If you reduce, when it comes to chemistry, if you're using chemistry terminology, to reduce carbon dioxide, that means you're adding electrons to it. In the process, that produces methane. Where do those electrons come from? Well, it comes from the NADH, right? The electron carrier. Technically, that's called oxidizing. But I'm glad I included that because I forget what your textbook looks like. But if you're reading your textbook, and they keep using the term reduce and oxidize, now you know what that means, right? Reduce just means to add electrons to it. Oxidize means to take it away. Anyway, those are methanogens that do that. I'm not going to ask you questions about that on the exam. There's also sulfate-producing microorganisms that reduce sulfate, right? So add electrons to it to make something called hydrogen sulfide. So again, I'm only including this because the textbook does too. I don't plan on asking any questions like that. The point here is that single-celled organisms can do different types of fermentation, and the two big ones that you need to know that I've taught you so far are lactic acid fermentation and methanol. All right, the next word for attendance is KFC. It was on this day that they opened their first franchise. I don't remember what year. And I'm not talking about the first restaurant, but the first franchise. Right? So the first time they had a new. Anybody guess where the first franchise was? The state. So obviously the first KFC was in Kentucky. I think Corbin, Kentucky. Anybody guess where the first franchise was? All right, I'll put it on the exam. I'm just kidding. It's Utah. A little bit of trivia for you there. Utah is the first place. Anyway, let's talk more about fermentation of bacteria. Like I mentioned earlier, some bacteria can use oxygen. Some bacteria have to use oxygen. Some bacteria can't use oxygen at all, right? Like they can't survive in the presence of oxygen. So these are what we're talking about here. So some bacteria, facultatively anaerobic, they switch between aerobic respiration and fermentation based on oxygen availability. That's a really long-winded way of saying, as far as this conversation is concerned, that basically like the yeast I taught you about, right? Yeast, you give them oxygen, they're doing cellular respiration. Take away oxygen, they're doing fermentation. I will mention this though. Hopefully you're a thinker. It's a little bit early in the morning, so hopefully this may not have popped in your head. But hopefully you remember when we talk about bacteria, those are prokaryotes, which means they don't have organelles, which means they don't have mitochondria. And I just told you about, electron transport chain, the citric acid cycle, and how that happens in mitochondria. But bacteria don't have that. So if you want to look it up for extra credit, because you don't need to know it for the exam, how do bacteria do cellular respiration when there's oxygen, right? Because they don't have a mitochondria. Anyway, those are the facultatively anaerobics. Then we have some obligate anaerobes, which means those are the ones that are unable to survive in the presence of oxygen. So again, some of them, they're flexible. They can go either way. Some of them, they have to have no oxygen. Anyway, again, this is just in the textbook, so that's why I'm including it. I'm probably not going to ask you any questions about this on the exam either. Do you have any questions about this slide? All right, here's a little, review sort of. Fermentation characteristics. All of it produces gas. All the different fermentation that I taught you about, other than the one that produces lactic acid, it produces gas. That's also not going to be a test question. Just so you know, also not going to be on the exam. A lot of times, this is how labs can identify what kind of bacteria they're dealing with. They can figure out what kind of gas it's producing. Is it producing hydrogen sulfide? Is it producing methane? Is it producing carbon dioxide? And then based on that, they can say, all right, well, it's definitely this group of bacteria. You might not be able to identify the species that way, but you can at least figure out the group it's in. So again, the importance of fermentation is to be able to ensure a steady supply of NAD+, right? The electron carriers. That's the purpose of fermentation. So we have to get rid of those electrons so we can keep stripping them away. So we need that NAD+. And obviously, if you didn't have that, you wouldn't have ATP production, right? And then, again, that would cause cellular death. So any questions? That's the last slide talking about fermentation. Questions about that? All right, and once again, for the exam, when we're talking about fermentation, I'm mostly concerned with the two different types. There's other types, but remember, the two you need to know about are lactic acid fermentation and alcohol fermentation. And it's all about stripping away those electrons and then adding them back to the pyruvate. And then depending on what type of fermentation it is, depends on whether you get lactic acid or alcohol, carbon dioxide, methane, stuff like that. But the idea there is no matter what, you don't have oxygen, so you have to put those electrons in. All right, so if there's no more questions, we'll talk about the last portion of this chapter. That would be nice if we could finish the last portion and jump into the next chapter, because this next chapter is about photosynthesis, and obviously the lab that we're about to talk about, the lab experiments are going to be about photosynthesis and respiration. So I'd at least like to get you introduced. But anyway, like I keep saying, as far as the exam is concerned, it's all about glucose, right? Break down glucose in the presence of oxygen. Break down glucose to make pyruvate. Glucose, blah, 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 blah. But how many of you have eaten in the past 24 hours? Okay, good. How many of you had nothing but glucose? Did you just get a spoon of glucose? Or did you have other stuff? Think back to the molecular, biological molecules. Is it possible maybe you've had some protein, some other carbohydrates, and some fats, right? You eat other things, and that's what this is about. 4.5, connections to other metabolic pathways. By the end of the section, you'll be able to discuss the way in which carbohydrate, metabolic pathways, glycolysis, and the citric acid cycle interrelate with protein and lipid metabolic pathways. It's a really long-winded way of saying you're going to learn how we break down other foods other than glucose in this process. And then at the end, you'll be able to explain why metabolic pathways are not considered closed systems. This section, we're going to talk about it. As far as the exam is concerned, it's very unimportant because I'm not really going to get into it. Just to keep things simple for this 100 level class, for the exam, we're just focused on glucose. Even when we talk about photosynthesis is how we create the glucose to begin with. But it also creates other things. And when we get there, for the sake of the exam, we're only going to focus on glucose. But anyway, cellular respiration is actually quite versatile, right? So, because again, we burn other things. We don't eat just glucose. Matter of fact, I would really, well, maybe some of you athletes did, but I was about to say, I bet most of you probably haven't had any straight glucose in the past 24 hours, unless maybe you had some sports drinks. You may have had some sugar, which is sucrose, but the chances of you having straight up glucose, probably pretty slim. So anyway, the next slide, I want to show you diagrams, some of the metabolic routes for carbs, fats, and proteins. For extra credit, notice I said carbs, fats, and proteins, but when you eat plants, you eat fungi, and you eat animals, obviously you're eating DNA too. So I'm not mentioning that, your textbook doesn't mention that, but for extra credit, you can look that up. What happens to the DNA that you eat? Is it broken down? Do you break down the DNA from your food and then use it to make your own DNA? When you break it down, do you use that to make energy? Because like I said, what I'm about to tell you is about carbs, fats, and proteins. I'm not mentioning anything about nucleic acids. Anyway, here's a picture from your old exam. I like it. All right, do your old textbook. Again, there'll be definitely no regular questions about this, maybe an extra credit question. So I might say, all right, which of the three stages uses carbohydrates directly? The only one that uses carbohydrates directly is glycolysis, right? Because you have these carbs, whether they be monosaccharide or disaccharides or polysaccharides, either way, they're broken down into sugars, unless they're monosaccharides and they're already broken down. But those sugars, those are the things that go into glycolysis. So yeah, we keep talking about glucose this, glucose that, but it could also be lactose or maltose or any of those other monosaccharides that I taught you about, right? So those monosaccharides, those are the things that go into glycolysis. Now, when you eat fats, hopefully you remember this, and you do need to know this for the exam, just as a reminder. Wait, was that the last exam? That was the last exam. But anyway, you'll need to know for the final exam, so here it is again. Remember, a fat, when we're talking about triglycerides, right? Those are one glycerol molecule of 3 fatty acids. So when you eat them, your body breaks it down into those parts. You've got your glycerol and your fatty acids. The glycerol is used in glycolysis, and the fatty acids are used in this process that was right before the citric acid cycle, right, where we turn the acetic acid into acetyl-CoA, and that's the thing we went into the citric acid cycle. And then, of course, you got proteins. Proteins are a little bit more complicated. Remember, there's 20 different types of amino acids, and they have all kinds of different shapes and sizes and even different atoms attached to the molecules, right? So remember the amino acid, well, you don't need to know it anymore, it's past this. But anyway, all those 20 different types of amino acids, when you break them down, depending on which one it is, some of them can go into glycolysis. Some of them can go to this process, the acetyl-CoA process. Some of them can go directly to the citric acid cycle. Again, I just talked a lot about that. But once again, I'll remind you, there'll be no regular questions about that on the exam. Might be some extra credit questions. Some good extra credit questions for this 100 level pass would be like the superlatives, right? So I might say, you know, what between carbohydrates, fats, and proteins, which is the one that can do, you know, any of these things, the glycolysis, the acetyl-CoA, the citric acid cycle, that would be proteins. And if I said which of the three between carbohydrates, fats, and proteins can only go into one of the three processes, that would be carbohydrates, right? So carbohydrates, very specific. Proteins, they do all kinds of stuff. And then fats, kind of in between. So any questions about this slide? All right. The next word for attendance, sushi. And I am going to pull down the next PowerPoint. While I'm getting this, go ahead and tell you, I recommend learning photosynthesis, or excuse me, respiration before you learn photosynthesis. Make sure you're really familiar with respiration because, sorry, I'm trying to download this here. There we go. Because, and this is a good way to put this, photosynthesis is basically the opposite of respiration. You'll see what I mean once we start talking about it. So if you're really familiar with respiration, then when you're learning photosynthesis, it should make it easier because you're just like, oh yeah, this is just the opposite of what I learned previously. Let's see here. We got the right one. Oh, okay. Bear with me. Sorry, that didn't work. That didn't work either. Oh, we might have an issue. Interesting. Looking at it right here. Download. Myself download. There we go. Might be on something funny. Yes, there we go. Okay. Here we go. Photosynthesis. Again, you know, a lot of my PowerPoints are based on, well, they were made based on the old textbook, and then when I got the new textbook, I changed them up accordingly. But some of the stuff I kept from the old textbook, including the beginning of the chapter where we always talk about, hey, why does this chapter matter for your everyday life? So why photosynthesis matters. This is from the old textbook. It's verbatim. Do you like to eat? We humans can trace every morsel of our food back to plants by capturing the energy of sunlight and using it to create organic materials. Plants performing photosynthesis feed the world. So obviously, even if you're eating animals, well, you're eating animals that ate plants. Even if you're eating fungi, right? Fungi are decomposers, so they are eating plants that used to live. So eventually, when you go back far enough, all this energy and the food came from the plants. Then we have these pictures. I'm trying to remind myself what this is. Yeah, I won't even talk about that. Let's just jump into it. We're running a little bit short on time. Got about 6 minutes left. So this chapter is broken down into three major parts. The basics of photosynthesis, followed by light reactions, followed by Calvin cycle. Now, remember when I was talking about respiration, I said a majority of the questions for that portion of the exam are going to be about those three stages of respiration, right? Which one produces this? Which one uses that? Which one does this? Which one does that? It's the same thing for photosynthesis, except photosynthesis is going to be a little bit easier because there's only two stages, light reactions and Calvin cycle. All right. Oh yeah, speaking of which, it's a good time to tell you this test question, but people get this one wrong and it breaks my heart. So again, you should memorize the big picture before you start memorizing the small picture, all the details. Hopefully by now you're very familiar with respiration or familiar enough to know the purpose of respiration is to use glucose to make ATP. That'll be a test question. And then the opposite is true here. The purpose of photosynthesis is to create glucose. So there will be a test question. where it basically says which one uses glucose, photosynthesis or respiration. It's respiration uses it, right? Someone's going to get it wrong, and I'll be sad about it, because that's like the easiest question. Then, of course, the opposite question will be which one produces glucose, and the answer is photosynthesis. So let's jump into it. So remember, you had to have some background information before we really jumped into respiration. Same here, you're going to need a little bit of background information before we jump into photosynthesis. So here we go, let's talk about it. Photosynthesis is used by autotrophs. If you don't know what autotrophs are, photoautotrophs. Those are basically organisms that use light to produce organic material. So that includes plants, algae, and some bacteria. Right? Some bacteria do photosynthesis. I'm probably not going to ask you that, but there you go. What happens is photosynthesis is transforming light energy into chemical energy. And we use that, and I'm definitely not going to ask you any of these bullet points. This is kind of an introduction to why this is important to you. But we use that chemical energy to make food, to make clothing, to make wood, make energy, right? So this is what photosynthesis does. And remember, this whole exam is basically about that transformation of energy from one form to another. When we're talking about respiration, we were talking about going from chemical energy to chemical energy, right? The chemical energy stored in the bonds of glucose were eventually translated into the chemical energy stored in the bonds of ATP. Here, we're talking about transforming light energy into chemical energy. So here's some pictures of what we just talked about. In case you haven't seen it, I'm sure you guys have. Those things are called plants on the left. Specifically, you got some trees and some flowers. Right here in the middle, you've got some photosynthetic protists, basically like different types of algae. That's kelp, but you need to know that. And there's some pictures of some photosynthetic bacteria. Anyway, here we go. Let's talk about chloroplast. So remember when we're talking about respiration, the shape and size, or not the size, but the structure of the mitochondria was important for respiration, right? Because we pump those hydrogen ions against the concentration gradient, and they were stuck between the inner and outer membrane, and then they had to diffuse back down, right? So if it didn't have that double membrane, it wouldn't work. So here we go again. We're going to talk about an organelle. Its shape was very important, its structure. Those are called chloroplasts. But that'll be a test question too. Someone's going to miss it too. Which of the two uses chloroplast? Photosynthesis or respiration? Photosynthesis. Which of the two uses mitochondria? Photosynthesis or respiration? Respiration. Someone's going to miss it. Anyway, what are chloroplasts? These are organelles. This is where that photosynthesis happens. These are the things that are absorbing the light energy. And not that I'm going to ask you this part, but they are mostly found in the interior leaves of the cell. And you'll see a picture that'll show. That'll make more sense when I show it to you. But it doesn't matter anyway for the exam because I can ask you that. But basically, I put a little star there to remind myself, to remind you. When you see a tree or a plant, all the green parts have photosynthesis. They have photosynthesized. So if it's green, that means it has chloroplasts in it. So even though we're talking about most of them are found on the interior leaves of the cells and leaves, they can be found elsewhere. That's just for you to know. I'm not going to ask you about that. But one of the textbooks, either the new one or the old one, does point out that you need to know this. That there are about 300 million chloroplasts in a piece about the size of a nickel. So if you got like a nickel and looked at a leaf, if you could zoom in, you would see there's about 300 million of them. But anyway, that's what chloroplasts are. I'm sure you've heard that term before. I'm sure you've heard chlorophyll before. Does everybody know what chlorophyll is? I'm sure you all know, but does everybody know how to describe it now that I'm putting you on the spot? First of all, it's green, in case you didn't know. And it's just the pigment. It's the pigment that's doing the light absorption, right? So the chloroplast is the organelle where it all happens. It's the chlorophyll is the pigment within the chloroplast that actually absorbs that light energy. We're going to talk about how that works, how it absorbs light energy. And obviously, because of that, it plays a central role in converting solar energy to chemical energy. And now we are done until 9 a.m. So I will see you guys at 9. If you guys decide to stay here, we'll escape out and say goodbye.